For the extraction of DNA, 700 microliters of blood was taken and mixed with 800 microliters of water, then centrifuged at 13,400 rpm for 1 minute. The supernatant was discarded and 1 milliliter of water was added again. Again, the sample was centrifuged at 13,400 rpm for 1 minute, and the supernatant was discarded. Subsequently, sodium acetate, SDS, and proteinase K were added and the sample was vortexed. The sample was incubated at 13.7 Celsius degrees for 1 hour and spin was performed. A mixture of phenol, chloroform, and isoamyl alcohol was added and finally centrifuged at 13,500 rpm for 1 minute. For the extraction of RNA from E. coli, the bacterial sample was first thawed and centrifuged to obtain the pellet, and resuspended in a test buffer. The sample was incubated for 10 minutes at 37 Celsius degrees and then at room temperature for 1 minute. 600 microliters of trisol was added and incubated at room temperature for 5 minutes. Subsequently, 160 microliters of chloroform was added and centrifuged at 12,000 rpm for 15 minutes at 4 Celsius degrees, and the supernatant was discarded. 1 milliliter of 70% of ethanol was added. Finally, the inverse tube was placed on absorbent paper for 5 minutes and resuspended in molecular biology grade water. For the measurement of the concentration of the samples by spectrophotometry, dilution of DNA and RNA samples with molecular biology grade water was performed and the wavelength was programmed at 260 nanometers and 180. A cell with molecular biology grade water was used as a target. In addition, the analysis was performed by agarose gel electrophoresis.
For the analysis of the samples obtained from the extraction of human genomic DNA and the extraction of RNA from the bacterium Escherichia coli, the observances of the samples were measured at a wavelength of 260 nanometers to verify the concentration of nucleic acid and at a wavelength of 280 nanometers to determine if there are proteins or another contaminating compounds present in the sample. To complement the analysis of the quality of the samples, an electrophoresis was performed. 0.8% agarose gel with TBE buffer 1x, providing a power of 110 volts for 60 minutes. In Figure 1, the results obtained in agarose gel electrophoresis can be observed. In lanes 2 and 3, the loads of human genomic DNA are shown. In lane 2, a defined band is not observed and a smear is observed along the lane. In lane 3, you can see a band with little definition, with an approximate size of 10 kb. You can also see what could be a runoff from the sample in lane 2. In lane 4 and 5, you can see 4 bands. The widest ones are between 0.050 and 0.150 kb in size, but have little definition. The 2 bands that follow are more defined than the previous one, but they have diffusion. They have an approximate size of 0.766 and 1.150 kb. In the upper part of lanes 4 and 5, a diffuse band with an approximate size of 10 kb is observed. In Table 1, The results obtained from reading the samples in the spectrophotometer at a wavelength of 260 and 280 are summarized. The concentration of nucleic acid was calculated with the help of the next equation. And the conversion to obtain the concentration in nanograms per microliter, in both cases abnormal values of A260 between A280 were obtained, which indicates an insufficient quality of the samples obtained from the extraction.
While in the bacterial cells the nucleic acids remain in the cytoplasm, they are constrained to a nuclear structure in eukaryotes. Cell lysis methods take into account such location to degrade additional structures that enclose nucleic acids. That is the reason we have employed SDS, which, due to its chemical structure, is a deterrent to lyse human leukocytes, forming micelles with the lipids that shape their membrane. Proteinase K is also playing a role in disrupting both membranes, the nuclear and the cellular membrane, and the chromatin. To lyse a bacterial cell, we used a chemical method relying on osmotic equilibrium by supplying high concentrations of sucrose, which osmotic shock eventually breaks the cell wall. Despite the type of cell, the steps of nucleic acid extraction remain the same. To select the correct nucleic acid molecule, DNA or RNA, some reagents are preferred. This selection can also be addressed by the pH adjustment of phenol solution. DNA and RNA are dissolved in the aqueous phase when adjusting the pH of phenol to neutral or slightly alkaline. But then, when adjusting it to acidic, only RNA remains soluble in the aqueous phase. Commercial versions are available to favor this selection. Trisol contains guanidine thiocyanate, sodium acetate, phenol, and chloroform, which allows sequential precipitation of RNA, DNA, and proteins. Further, cell walls contain polysaccharides that need to be eliminated from the sample. CTAP is a reagent that allows the precipitation of both nucleic acids and polysaccharides to finally retrieve the soluble nucleic acids in the presence of high salt solutions. But in this case, this is not necessary because we are not dealing with cellulose. Regarding the purity of nucleic acids, the 260 to over 280 index was low, because a value of 1.8 or similar is considered to confirm a pure DNA sample, or at least that it is adequate to use in further procedures. A value of 1.25 indicates the presence of contaminating proteins in the genomic DNA sample and possible contamination with phenols. After the agarose gel electrophoresis, it becomes clear the presence of such molecules at the top of the lane 2 and 3. Perhaps better storing conditions and nuclease inactivation will improve the quality of the samples, as they are slightly degraded. In the RNA purity measurement, a very high value was obtained. Despite the calculated index of 7.0, proteins are observed at the top of the lanes 4 and 5, at quantities similar to the genomic DNA samples. These contaminations could be caused by the use of dirty tips and the necessity of extra phenolic extraction. Despite lanes 2 and 3 contain the same sample, lane 2 shows an abnormal band. This happens probably because during the preparation of the electrophoretic chamber, some sample was accidentally spilled. Moreover, the voltage could be reduced to resolve the bands better. The ribosomal subunits 23S and 16S are clearly visible in lanes 4 and 5, though they are degraded. The extraction procedure was effective despite some active nucleases remaining. Thus, this sample of genomic DNA is not adequate to use in techniques like PCR because it contains contaminants that may interact directly with DNA chains or with thermostable polymerases, inhibiting amplification. Enzyme inhibition may occur as well by the presence of magnesium-reducing agents, affecting the activity of the restriction enzymes and polymerases in sequencing.
Design a protocol for the extraction of nucleic acids. Three things need to be taken into consideration: the type of genetic material that wants to be retrieved, due to the difference according to their stability, the cells where the genetic material is located, since their structure according to membrane and cell wall will vary if the cells are from plants, animals, bacteria, etc., and the conditions of the sample, since the storage period may affect the state of the nucleic acids. The electrophoresis and spectrophotometry are two methodologies that, used together, provide an order characterization of the quality of the DNA or RNA extracted. Through electrophoresis, the contamination of the sample by salts and proteins, the degradation of the genetic material, and the type of nucleic acid extracted is determined. The spectrophotometric analysis allows the quantification of the sample and the evaluation of its contamination level.